Shut that down just for a minute. Come on. No, it's not going now. There we go. Share the screen. Come back to the syllabus. So today we're covering lab 4. This will be on stains. You'll note we do not have a lab next Thursday. I will be here to answer questions though. So let's go to lab 4. Lab 4 is on staining techniques. We're going to talk about simple, differential, and special stains. You should read the class textbook before doing the lab, and there's a fair amount of reading, but each section is just very short. But there's a large number to read because we have different types of cells that we stain. Anyways, read about the size and shape and the arrangement of bacterial cells. Read about the bacteria cell wall. Read about what is a simple stain. Read what's a differential stain. And you should note that there are 2 differential stains we'll talk about, the Gram stain and the acid-fast stain. You should also know that there's a 3rd type of stains that we call special stains. And we'll talk about negative staining for capsules, endospore staining, and then flagella staining or flagellar stain. There are some supplemental materials in this lab. You're going to be working with video clips, watching little movies, and we have a virtual lab today. The learning objectives: be able to prepare bacterial smears for staining. Students often don't know what I'm talking about when I'm talking about making a bacterial smear. You make a bacterial smear before you do the staining. So bacterial smear is not doing the staining. It's putting the bacteria on the slide. Understand the principles and the procedures used when performing a simple stain, a differential stain, and then the special stain. Be able to interpret the results for a simple stain, a differential stain, and a special stain. And be able to define the following terms. What's a simple stain, a differential stain, a special stain? What's a coccus? What's a bacillus? What's a sporillum? What is peptidoglycan? What are gram-positive cells, gram-negative cells? What's an acid-fast cell or acid-fast staining cell? What's a primary stain? What's a mordant? What's a decolorizer? What is a counter stain? What's a capsule? What is an endospore? And what is a flagella? Any questions? All right. As there's no questions, let's continue. Because of their extremely small size, bacteria are transparent. And they're also mostly water, so that tends to make them transparent. Therefore, it is necessary to stain them before you can look at them under the microscope. Preparing a bacteria smear is just adding a drop of sterile water on a clean slide, adding the bacteria to the drop, and you only add a little bit, by the way. And then you smear that drop all over the surface of the slide. So spread it around. You then allow the slide to air dry, and then you heat fix. The reason for doing these 2 are to get the bacterial cells to stick and attach to the glass. If you do not allow the slide or the bacteria smear to air dry and heat fix, then the bacteria will not attach to the glass, and then the water washing or other steps of the smear will wash away the cells. Any question about that? A simple stain is when you simply use one dye to stain the cells. And note here that it's saying a basic dye. That's not really correct. It can be a basic or an acidic dye, and it's still a simple stain. The simple stain is just using one dye. The differential stain usually uses 2 or more dyes and provides additional information about the cell. that a simple stain cannot give you. Like when you use a simple stain, all the cells will be stained one color. If you use a differential stain, depending on what the cells are, but you can get the cells to stain differently, meaning a different color. The Gram stain is the primary differential stain we'll talk about. And then lastly, there are special stains, and they're used to look at special structures and cells. When we're talking about cells, it's good to learn the terminology for the shape of cells when they stick together. And this isn't showing it here, but if we have only single bacilli or a single cocci, we would call it a singlet or a single cocci. It's not shown here, although it is shown for the bacilli here. You can also have cocci that are strung together in a line or, I don't know, a bead, and that we call streptococci. More than two hooked together in a line. For bacilli, they have to be lengthwise to be called streptobacilli, and you can think of that as a chain of hot dogs linked together on the long link. I guess I have to mention that, on the long side of the hot dog. You can have the cocci hooked together in a cluster, and the cluster sort of looks like a cluster of grapes, and that is called staphylococci. And that's actually what staphylococci translates into, cocci in the shape of a cluster of grapes, staphylococci. That's what that word means. You can also have tetrads of cocci, and you can have sarxinia. I think I've got that shown down here. No, I don't. You have sarxinia of cocci, and that's 8 cells hooked together, so 2 tetrads stuck together. And then you can have diplococci, 2 cocci hooked together. Okay, you can have diplobacilli and streptobacilli. You do not have staphylobacilli, and you do not have tetrads of bacilli. Any question about any of that? With spirilla, all you have are singlet spirillas, meaning they only form singlets. They do not stick together. You should know the difference between a bacilli and a cocci. Cocci is a round ball shape. Bacilli is I don't know, a longer cell. It can have pointed ends. It can have blunt ends shown here, or it can be rounded ends, sort of like that one there. Okay, and I would not use the term spirilla bacilli. I would only call it spirilla. Okay, a bacilli is this. A spirilla is like an S-type shape. Most cells are monomorphic. They take on one shape. All cells are cocci, but you can get cells which are pleomorphic, and they take on different shapes, as shown here. You can also have coccobacilli, and that's a cell in between the shape of a cocci and a bacilli. There are some other shapes, but we're not going to talk about it in the lab. I will briefly mention a few of them in the lecture. There's star-shaped cells and square-shaped cells. But these are the common ones. The spirilla, the bacilli, and the cocci are the 3 most common. And most of the others are a shape derived on that. Like the coccobacilli is definitely a shape derived from a cocci and a bacilli. All right, oops, that looks like my lecture finished. So I've got the file now to post it, but I'll do that after I'm done talking in the lab.
I'm sorry, can we also get a PowerPoint for chapter 2, please?
Remind me after I'm done giving the lab, talking the lab, and I'll do that in the
Thank
lab.
you.
Okay. Remind me or I will forget. Gram staining. So we mentioned that there are simple stains that uses only one basic dye. All the cells will be stained the same. You can have a differential cell, which the cells can take a different shape depending on what the cells are, not a different shape, can take a different color depending on what the cells are. If they're different and the differential stain can differentiate them, they'll take a different color. The Gram stain is a differential stain and it was invented in 1884 by Hans Christian Gram. So whenever you write the Gram stain, it should be with a capital G because it's named after Dr. Gram. He used it as a differential stain to differentiate between a person's cells and a bacterial infection. But microbiologists started taking his Gram stain and started using it, and they noticed that it can differentiate between bacteria. And that is why it is the most commonly used stain in microbiology, because it can differentiate between bacteria. It differentiates on their cell wall. I'll talk a little bit more about that. In the Gram stain, crystal violet is the first dye used, so we call it the primary dye. Bacteria retain the crystal violet and they will take up the purple color. And if they retain that, we call them gram-positive cells, where they stay purple. But bacteria can lose the purple color, and that they lose it with the decolorizing step. And these cells we call gram-negative. We then color these cells which lost the purple color with safranin, and then they appear pink. So if we were to do the Gram stain on a mixed culture of cells, you might get something looking like that, where some cells would be gram-positive, and these are gram-positive bacillus. There's a gram-positive rod right there. Another gram-positive rod or coccobacilli. And there's a gram-negative rod right there. There's gram-negative cocci right here. And there's a gram-negative spirilla right here and right there. So that's a typical Gram stain. Gram reactions are based on the bacterial cell wall's ability to retain the crystal violet. Gram-positive cells contain thick peptidoglycan. I guess I should say bacteria, because that's what I'm talking about. Gram-negative cells, on the other hand, have a thin peptidoglycan layer, and it's not thick enough to retain the crystal violet, and so it washes out. So when we look at the gram-positive cell, we'll notice it has a plasma membrane right here, and then a very thick peptidoglycan layer being the cell wall, and peptidoglycan is the major molecule in the cell wall. In the gram-negative cell, peptidoglycan is the main molecule in the cell wall, but it's a much thinner layer. And there are other molecules that we see in the gram-negative cell, mainly the lipids and the carbohydrates of the outer membrane of the cell wall. Okay, so there's the plasma membrane, and this part here being the cell wall. And this is a gram-negative cell. It's got a thinner peptidoglycan, and it has other molecules in it, mainly the lipids. A mordant is a compound that simply intensifies the primary dye. And in the Gram stain, iodine is used as a mordant. It doesn't really color the cell. It intensifies the crystal violet. What happens is the iodine complexes with the crystal violet, makes a larger molecule. And so that makes the crystal violet stain the cell a little more intensely, gives it a little browner color, meaning a little more purple color, and prevents it from washing out the decolorizing step because it's a larger molecule. So it helps it prevents it from washing out. All right, so typically we have bacteria which either are gram positive or gram negative. I do need to warn you that there are some cells which are called gram variable, where the cells lose their ability to hold on to the crystal violet. And these are usually the bacteria in the family Mycobacterium. They have a slightly different cell wall. We'll talk about that much later. Sometimes when you stain the mycobacteria, you may get this color where it looks like some are gram positive, some are gram negative, even though there's only one species here. What do you notice about this cell in particular, and that cell a little less, that cell there, compared to that cell and that cell and this cell? What do you notice between this cell and that cell, this cell and that cell? Come on, somebody's got to see it. All right, I'm not getting any answers, but think about it because I'm not going to tell you. You guys have to supply some answers when I ask questions. Usually what happens when the mycobacterium stain this way, I have had it happen a couple of times with a few students, and I asked them, how old were the cells before you stained them with the gram stain? And almost always the students have told me, oh, these cells were older than 2 weeks old. For the gram stain, you need to use freshly grown cells or the stain may not work correctly. And when they were using 2-week-old cells, they were seeing this. And the reason why they were seeing this is because some of the cells were too old and they were dying. And the old and the dying cells stain gram negatively, and the fresh and the newer derived cells are staining gram positively. If they would have used the cells the way they should have, meaning to do the gram stain, you should only do it on cells that are 2 days old or less. So recently grown cells, they will all stain gram positively. Okay, so did anyone see a difference between this cell and that cell, or this cell and that cell? Nobody sees a difference?
Yes.
What's the difference between, besides the color? All right. You guys will have to figure it out on your own.
Is there no difference?
No, there's a difference. You don't see a difference between this cell here
Oh, just the color?
and this cell here and that cell? No, a difference in something other than the color. That
The
cell
shape.
here,
The shape, the texture.
the texture is correct. Not the shape, they are both bacilli, okay? But this cell here, and that cell here, and that cell here, these are all ghostly cells. That cell is very intense. Same with that one. So these cells have a very distinct boundary, very sharp boundary between the cell and the end of the cell. Whereas that cell and this cell and this cell, they don't have as sharp of a boundary. This one in particular, you can see that that cell looks like it's almost broken. And what happened was this cell is a dead cell. So was that one and that one. And that's why the cell is ghostly. Not all of the cell is there. And the dead cells, because not all the cell is there, it's not staining correctly with the gram stain. And that's why they're gram negatively stained. Whereas a new cell, this one here and that one, it's living, vibrant, and you can look at the cell and be certain that there's a distinction between what's inside the cell and what's outside the cell. Whereas that one and this one and that one, you're not entirely certain exactly where the cell even ends and where it begins. That is a dead cell, it's disintegrating. And the point is, when you do the gram stain, do it on freshly grown cells, and then you won't see the gram variable result. I've never done it on Mycobacterium where I didn't get them to be gram positive, but I haven't tested all the members of the family Mycobacterium. In fact, I think I've only tested 2 species of the family Mycobacterium. All right, let's move down. So to prevent gram variable results, avoid using old cells. Cells should only be 2 days old or younger. Prepare thin smears. A thick smear has a problem because the cells are lined up on top of each other, and it's hard getting the dye in the very bottom cell. It's also hard if the decolorizing agent is supposed to remove the stain on the very bottom cell. It's harder to remove that stain. So make it a thin smear. Ideally, a bacterial smear should only be 1 cell layer thick. And then heat fix the bacteria only after the smear is air dry. If you heat fix and the cells are still wet, you'll get boiling in the cell, and that'll put holes in the membrane of the cell as well as the cell wall. And then that can allow the dye to enter when it shouldn't. And then avoid overheating the cells, avoid overheat fixing. Okay. Some other things that help will be using only fresh reagents. Do not over decolorize. That we'll talk a little bit more. This, I've never seen this being a problem. Rinsing too long with water. I can't imagine that removing crystal violet when it shouldn't remove it. But if you over decolorize, you can remove the crystal violet when you should not. And do not under decolorize either, because you may not pull out the crystal violet when you should if you under decolorize. All right. Another differential stain is the acid-fast stain. Some bacteria, particularly the family Mycobacterium in Nocardia, they have waxy substance in their cell walls. That waxy substance is mycolic acid. And the mycolic acid prevents water-based substances from going into the cell. And the gram stain is water soluble. So it doesn't stain the cells as well. These cells take up the acid-fast stain, so we call them acid-fast bacteria. An acid-fast cell stains pink from taking up the stain carbol fuchsin. I don't even have it here, but that's the stain. And the acid-fast negative cells do not retain the carbofusion stain, and they will either appear bluish or green, depending on what the counter stain is. And this actually looks a little turquoise, so blue-green right there. Oops, too far. Let me get that back. Having a hard time. Oh, come on. That's close. About right there. All right, so that's the differential stains. Like I said, there are two you'll need to know about, the Graham stain, the acid-fast stain. There are also special stains, and these are stains that specially stain specific components of the cells. We can use a negative stain as a special stain, and this will stain the glycocalyx of cells. The glycocalyx is something the cell secretes, and it's attached to the cell wall. It's external to the cell. If you can see it under the light microscope, the glycocalyx is we call it a capsule. If you cannot see it under the light microscope, and you have to look at it under the electron microscope to see it, this glycocalyx we call a slime layer. So there's the capsule, the cell wall. Anyways, when you stain for the capsule or glycocalyx, it uses a stain that usually stains the background of the cell and the cell, but it does not stain the capsule or the glycocalyx. A capsule is important for cells because it can be associated with the organism's virulence or assist in its pathogenicity, meaning a cell that has a capsule is more virulent or harsher on the patient. It's more pathogenic than a cell without the capsule. And you can read about different cells that have a capsule. The capsule also makes the cells look mucoid. The reason why these colonies look mucoid is because they have a capsule. So here's a special stain, staining the cells. And this actually isn't in India ink, I can tell, because it's too dark of the cell. India ink stains the background very darkly, and then the cells very lightly. This is staining both the background and especially the cells very darkly. That's why I know it's not India ink. But this cell has a capsule, and you can see the capsule does not stain. Any question about that? All right, another, go on, go ahead.
What about the other cells? I also see capsules around the other cells.
Yeah, this doesn't look like a bacillus, so that can't be Klebsiella pneumoniae, because that looks like a cocci. I don't know what these cells are, it doesn't say. Okay, and I got this from a picture from somebody, and like I said, the picture has that this is India ink. I can look at this and tell you this is not India ink, because India ink stains the background very darkly, and it lightly stains the cells. It does stain the cell, but not this darkly. Endospores, the endospore stain is another special stain. Endospores are a metabolically inactive cell. And what happens is when the cell comes to an environment where the cell will die in that environment, the cell starts making an endospore. And then the endospore is inactive and a hardy structure. And then the cell can continue to live in this harsh environmental condition. And it'll just survive for a long period of time. When the environment changes and becomes favorable for the cell to grow, the endospore will germinate and the bacteria will return to its normal metabolically active vegetative cell. And a vegetative state or vegetative cell is a cell that is metabolically active in its normal state, and it's actively metabolizing and reproducing. Okay, an endospore is inactive, and its purpose is just to survive. This table is giving you some pathogenic endospore forming gram-positive bacilli. So some important species which form endospore are given here. I think you've all heard of Bacillus anthracis causing anthrax, and you've probably heard of food poisoning. Bacillus cereus is one of the bacteria that can cause food poisoning. There are actually many that can, but Bacillus cereus is one that forms an endospore and causes food poisoning. Botulism, I'm sure you've heard of. Tetani or tetanus, Clostridium tetani, forms an endospore. Clostridium perfringens also causes food poisoning. So there's 3 that form food poisoning and then also form endospores. And then I'm sure you've heard of C. diff, Clostridium difficile, which causes antibiotic-associated colitis. It also has an endospore, and that's why C. diff is so difficult to treat. It has an endospore. The endospore can survive the antibiotic treatment. And so you always have to treat a patient with antibiotics for at least 2 courses at 2 different times to get rid of C. diff. The first time you get rid of the vegetative cells, but not the endospores. And then you remove the patient from the antibiotic. The C. diff endospores then find the environment favorable to grow. And so they germinate. And if you get it right after they germinate, there will be no cells in that have endospores. And then you treat with antibiotic the second time. And then you can remove C. diff from the patient. All right, here's an endospore stain, and you can notice that this one is like a differential stain. It stains the cells differently. The endospores are staining green. And here we have an endospore inside a cell. There's a little bit of red cell up on this side and a larger portion of red cell on that side. The endospore is green in between. Here is an endospore that has been separated from the cell. Right here. All right, and we'll talk about in the lab, you'll read about the Schaefer-Fulton endospore stain. And that's what we're looking at here. Another special stain is the flagella stain. It stains the flagella, which are not normally seen when we stain the cells. I don't know if Klebsiella pneumonia has a flagella or not, but let's pretend this is anthrax. It has a flagella, or some E. coli have a flagella. Lots of cells have a flagella. And when we stain them, we don't normally see the flagella because the flagella is too small to be picked up by the dye. And what you need to do to stain the flagella, where is that? Right here. Is that you use something where you coat the flagella with many different coats, and then the flagella will show up. So they're long thread-like appendages, which are used for bacteria to be motile. If the bacteria can swim or is motile, it has a flagella. That is the only way that bacteria can swim or be motile in a liquid culture. because that's the only structure bacteria have for motility. This picture shows you the different way the flagella can be arranged. You don't need to know this, but there is a question in the lab about what is the flagella arrangement for the cells. And so you can look at this and get it. And let me warn you, here's the picture, that you can see, look at this, that this one is, and this one and this one are clearly peritrochus, but this one looks more amphitrochus, and this one might be even just one flagella. So that would be monotrochus. You can also have a few flagella on one end, lophotrochus, lophotrichus, and I don't know if I can find one. Actually, that looks like lophotrichus. When you look at the cells, what happens is the first flagella is made, and then there's only one. And then the cell will put down more, and it will be lophotrichus. And then it'll put down more, and it'll become amphitrichus. And then the species will put down more, and it'll be peritrichus. So when you talk about the flagellar arrangement, you look at the cells that have the highest number, because they've been around for a while. They've got their complete flagellas made. You don't report it for the ones that are still putting down the flagellas. Is that clear? All right, so know the terms. And then do the lab. You don't need to look at the references. Lab 4, while you're not physically going to perform this activities, you are expected to understand how to prepare bacterial smears for staining. You also should know how to make a bacterial smear. We got a movie on it, and then how to heat fix the smear after making the bacterial smear, and you heat fix after it's air dry. Read through the procedure for making a smear. Let me state that the #1 problem that students make when they actually make a bacterial smear themselves is they add too much cells. So you only mix a small amount of specimen in a single drop of water. If the organism already is in a liquid broth, all you add is a small drop of the broth. You then let it air dry, smear it around on the slide, and then let it air dry, and then you heat fix, let the slide cool, and now the bacterial smear is ready for staining. So that's what we do to make a bacterial smear. We do not do the stain for making a bacterial smear. A simple stain, you take the bacterial smear and then watch the video on making the simple stain. But essentially what you're doing is you're staining with a single dye, like methylene blue or crystal violet. All the cells will wash, will take on the same color. Okay. All right, so here's a smear of a simple stain. They're both purple. This is E. coli, and this is Staphylococcus aureus. Let me warn you that right here, that's 2 cells hooked together. Right there, that's at least 2 cells hooked together. This almost looks like it's a spirilla right there and right there, but that's because the cells are hooked together and right there. If you look at only the singlets right there and right there and right there and right there, and right there and right there, you'll note that the bacilli singlets are all, excuse me, the E. coli singlets are all bacilli. and then describe their morphology of E. coli and Staphylococcus aureus. Here we're seeing Staphylococcus aureus. There's E. coli. And answer the question, what's total magnification? Then do procedure 3 on the gram stain. We have a little video on doing the gram stain. So watch the video and then run the virtual lab. The virtual lab will take a little while to download. Up. This one we might not have. Let me check on the newer version. See if we do have a virtual lab. Yeah, we do have a virtual. Nope, it's the wrong one. So. Yeah, we have a virtual lab in this one. So let me scroll down to where we were. Come on, there it is. And I've got to open it from this one. Sorry, I haven't updated my own hyperlink. And the old hyperlink doesn't work because Flash no longer runs. Oh, good, this one loaded quickly. Continue, gram stain.
Hi, Lab Assistant. Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by some undesirable bacteria.
Let's go ahead and go down to the gram stain, unless that's the movie. That looks like it's a movie.
All yogurt has bacteria in it since unfortunately, undesirable bacteria like salmonella. This is what plain good yogurt looks like. Now take a look at this yogurt. This yogurt looks. Let's collect yogurt samples and prepare them for viewing under the microscope by doing a gram stain. In the gram stain, bacteria that stain a pink or reddish color like this are called gram-negative, and their presence means the yogurt is contaminated. Bacteria that are used to make. We are going to gram stain sample. All right, let's prepare our slides for gram staining. To prevent bacteria from contaminating our work area, we have wiped down the table with a 70% ethanol solution. We will also light a Bunsen burner to sterilize the inoculating loops we'll use later and help maintain a sterile working environment. You're going to use a striker to light the Bunsen burner. The gas is turned on low already. We need to dilute the yogurt before we put it on the microscope slide. We'll use a PBS liquid. Start by placing 1 milliliter of the diluent into a test tube using a pipette. This is an inoculation loop, a small loop of wire used to collect samples. We'll use it to place a small amount of yogurt into the test tube with the diluent. Start by sterilizing the inoculation loop in the flame of the Bunsen burner. Hold it.
Red.
After it has cooled down, use the loop to obtain a small. Place the yogurt sample into the previously measured diluent,
I'm using
PBS.
the mouse to get this virtual lab to run. I'm just going to end that here, So work on the virtual lab and then view the steps for performing a gram stain. First, you flood the smear with crystal violet. You leave it on for 1 minute. We have a little picture of that. Here it is. You then dump off the crystal violet and you do a water rinse. After every step, you do a water rinse. I have that on there. Yeah, every step's rinse with water. And then you flood the area with grams iodine. Iodine is a mordant. It intensifies the stain of the crystal violet. And then you dump off, leave that for 1 minute. And then you dump off the iodine and then do a water rinse. Then you apply the grams decolorizing agent. And this can be different agents. It's usually something like acetone and alcohol mixture, and it decolorizes the cells, pulling out the crystal violet iodine complex from the gram-negative cells, but not from the gram-positive cells if it's done correctly. You only apply this for about 5 seconds. The best way to do it is to watch while you're doing it. So here's your slide. Hopefully you can see this. Apply the decolorizing agent. And when the color stops running, stop applying the decolorizing agent. That's the best way to do it. You then immediately dump off the decolorizing agent and immediately rinse with water. The purpose why you need to be immediate at this step is if you leave the decolorizing agent on the slide, it'll continue acting. And if it's given enough time, it will pull the crystal violet out of gram-positive cells when it should not be pulled out of those cells. And then the cells will be, the gram-negative cells will be colorless. So this is called the differential step of the gram stain, where the gram-positive cells stain purple, the gram-negative cells are staining colorless. And then we'll apply safranin, which stains all the cells, but it makes the colorless cells stain pinkish. And so the gram-negative cells stain pinkish, and the gram-positive cells stain bluish or purplish. Any question about any of that? Oh, you leave the safranin on for a minute or 2 minutes. And then you do a water rinse after that. So after every step, you do a water rinse. So when you're describing the gram stain, all you need to do is say in one place or another, after every step, I do a water rinse. You don't have to say each step and then do a water rinse, do the next step, do a water rinse, etc. And then you can blot the slide with bibulous paper, put the slide inside, and then fold the bibulous paper over. Bibulous paper is very thick paper, almost like cardboard, a little thinner, and it's much more like paper, but it's very absorbent, and it will pick up the water. You do not scrape the slide onto the paper, but you can blot it, meaning, I don't know if you can see this, but put the paper on the slide, this is the slide, and then just blot it. That will remove enough of the water so that you can then observe the cells under the microscope, including under the 100x microscope with oil, meaning slight traces of water will not interfere with that. And then in this lab, we have pictures showing you E. coli stained under gram-negative cell and Staphylococcus aureus stained under the gram stain. Sorry, these are both done under the gram stain. And so describe that in the table. You need to also state what color Staphylococcus aureus was and E. coli was at each step of the gram stain. So fill that out. On procedure 4, we're going to look at the acid-fast stain. We have a little movie for you. The acid-fast stain we're going to talk about is the modified Kenyon acid-fast stain, which uses Kenyon carbol fuchsin. The primary stain, which is carbol fuchsin, the first stain, stains all the cells pinkish. The decolorizing agent removes the carbol fuchsin from the non-acid-fast staining cells, but not from the acid-fast staining cells. And then we use brilliant green in this stain, or this slide, this procedure, sorry, to stain the non-acid-fast stains, give them a color, and brilliant green is green in color. So with this procedure, the acid-fast cells will stain pinkish, and the non-acid-fast cells will stain greenish. With the Zeil-Nielsen method, you'll notice it's a different color, but the acid-fast stains stain pinkish, and then the non-acid-fast stain stains, well, kind of a bluish green or turquoise color. You do need to answer the questions here. Students seem to miss this question. Seen below are two smears at 1000x that are AFB stain, meaning acid-fast stain. I don't remember what the B stands for. Interpret whether the organisms denoted by the AFB positive or AFB negative. So where each arrow is, state whether it's AFB positive or AFB negative. That one's positive, that one's negative. And you can state, like on the left picture, the first arrow is whatever the second arrow is, or you could say the left arrow is and the right arrow is, or the top arrow is. Okay, so you need to write that down, what they are. That is a question. Interpret whether the organisms are AFB or AFB negative. I understand why that's a problem. And that is, there's no question there. That should have a question mark there. I'll put that there and hopefully someday I'll remember to upgrade it. All right, then we're going to look at special stains. The first, watch a video on the endospore stain. We also have a video on a negative stain. We're going to use a negative stain to stain the cells for cell capsules. This is a endospore stain. It's the Schaefer-Fulton stain. And you have to state what stain is being used as the primary stain, and ultimately what it dyes, what it colors. And says these are produced by some bacteria to aid in survival. List some examples of pathogenic endospore producing bacteria. You can get that from the lab manual. Shown above. One of the tables up here. Is that the one I want? Yes, some examples of pathogenic endospore producing bacteria. You can just list some or all of this table, okay? And then B, this stain goes to figure B. What stain is it? And it's coating a long whip-like appendages which aid in motility. So name the stain. State the flagella arrangement type in this cell here. And then list some examples of clinically significant bacteria that produce flagella. And you can find that in the lab module. And then stain C, this is a special stain, but what one is it? And it uses India ink or nigrosin and can be used to detect the presence of what, which is located exterior to the cell wall and can help prevent phagocytosis. Okay, and then list some pathogenic microorganisms that produce capsules, and you can get a list of those from the lab manual. And then do 2 case studies. The first case study, we give the information here, and then the X-ray, chest X-ray, and we'll note something funny here and something funny here. I should have a picture of a real chest X-ray. Wonder if I have that someplace else. And then we do a sputum smear and then stain it with the Zeal-Nielsen stain. And we're seeing 2 types of cells. And then give your most probable diagnosis. You need to name the species of bacteria which is causing the infection of this case study. All right. So name the species of bacteria, although it is true that this disease only has 1 species that causes it. So it's okay. In case study 2, we have another case where the information is here. And then give the most probable diagnosis. You can see the necrosis on the patient's foot, the gram stain of the bacteria causing this infection, and then the Schaefer-Fulton stain, staining something, and then name the species causing this infection. And you can get the species in the lab module. All right, any questions about the lab? If not, go ahead and get started on the lab. I will be here until 8 o'clock to answer questions. And I'm supposed to do something. What was it?
Can you please send us the PowerPoint for chapter 2?
Okay, I'll put the PowerPoint up online.
And then is the test opening tonight at 8?
Is what opening at 8?
The quizzes, are they opening today at 8?
Oh, yes. That reminds me, I need to do something. I've got to do something for the quiz before you guys get started. So let me get the PowerPoint right now. I'm not going to be able to take questions because I've got to do something with the quiz right away. All right, let's get the PowerPoint up.
I'll have to take it today because I work until 7:30 tomorrow. So by the time I get home, it's usually 8 o'clock.
No, you'll have to take it before going to work, or you won't, or tonight.
Yeah, I'll take it
I thought it was, yes, it is right here. So let's go to the week 3 lecture, week 2 lecture. There it is. Let me go ahead and put on week 5 of the lab. Oh, I've already got it up. Never mind. All right, let's go into the quiz. Quiz 1B. Oh. Oop, didn't want to do that. That didn't do that. Yep, it did this time. Come on. Oh, shoot.
And Professor, is this quiz only one?
Not now, not now. I'm trying to do something to make the quiz work. Give me Give me about 5 minutes, okay? And then you can ask your question. Sorry, but this needs to be up before 8 o'clock, so. I don't know. Oh, there it is. All right, who else do I need? Was I sharing that? That was a mistake.